Dear friends, good morning to all. Today we can discuss about types of enzyme inhibition or enzyme inhibitors. Enzyme inhibition, blocking an enzyme's activity by chemical molecules which resemble or mimic a particular enzyme substrates is called as enzyme inhibition. And the molecule is said to be inhibitor. Therefore, many therapeutic drugs are some type of enzyme inhibitor. Molecules that bind to enzymes and increase their activity are called as enzyme activators. The binding of an inhibitor can stop a substrate from entering the enzyme's active site or hindering the enzyme from catalyzing its reaction. The modes and types of inhibitors have been classified by their kinetic activities and site of action. Next one, types of inhibition. Reversible inhibition, another one is irreversible inhibition. First we will see about definition for reversible inhibition. Reversible inhibitors bind non-covalently. Non-covalently means either hydrogen bond or hydrophobic or interaction or ionic bonds. Different types of inhibition are produced depending on whether these inhibitors bind the enzyme or enzyme substrate complex or both. Next one is irreversible inhibition. Irreversible inhibitors usually reacts with the enzyme and changing it chemically. These inhibitors modify key enzyme residues needed for enzymatic activity. Types of reversible inhibition. First one is competitive, second one is non-competitive, third one is uncompetitive, fourth one is mixed type of inhibition. First one, reversible inhibitors, competitive inhibition. In competitive inhibition, the substrate and the inhibitor cannot bind to the enzyme at the same time. Here the substrate and inhibitor compete for binding to the enzyme's same active site. The reaction enzyme plus substrate to form enzyme substrate complex. Finally we can get the enzyme plus product. Otherwise the inhibitor bind to enzyme means enzyme inhibitor complex formed. Here no reaction takes place. The diagrammatic representation, enzyme which have the binding site, that is called active site. Substrate binding means enzyme substrate complex formed, or inhibitor binding means enzyme inhibitor complex formed. Rate of reaction is Km or Ki. Here competition takes place between the inhibitor as well as substrate. So this is called as competitive inhibition. Next one is under the reversible condition, non-competitive inhibition. Non-competitive inhibitors bind both the enzyme E plus I enzyme inhibitor and the enzyme substrate complex plus I enzyme substrate inhibitor complex formed. The inhibitor binds to a site other than the substrate site and is thus independent of the presence or absence of substrate. This action results in the conformational change in the protein that affects the catalytic step and hence decreases or eliminates the enzyme activity. The reaction enzyme plus substrate to form enzyme substrate complex. Finally, we can get the enzyme plus product. Two ways here reaction will takes place. And after formation of enzyme substrate complex, then inhibitor binds to form enzyme substrate inhibitor complex, or otherwise first enzyme reacts with the inhibitor to form enzyme inhibitor complex, then it will be forms the substrate to form enzyme substrate inhibitor complex. The diagrammatic representation, the enzyme which has the both side substrate and also inhibitor binding sites. Once substrate binding means next will inhibitor bind it, or first inhibitor binding means next substrate will binding it. Finally, we can get the enzyme substrate inhibitor complex. So here, so this form are called as non-competition. Third one is under the reversible category uncompetitive inhibition. In this uncompetitive inhibition, the inhibitor cannot bind to free enzyme, but this type of inhibitors only bind with the enzyme substrate complex. The enzyme substrate inhibitor complex thus formed is enzymatically inactive form. This type of inhibition is rare but may occur in multimeric enzyme reactions. Enzyme reacts with substrate to form enzyme substrate complex, normally which leads to enzyme plus products. This type of inhibitor only binds with the enzyme substrate complex. Finally, we can get the enzyme substrate inhibitor complex. Here, no reaction takes place. Diagrammatic representation, here also the enzyme which has the both binding sites, substrate as well as inhibitor. First substrate binds to enzyme to form the enzyme substrate complex. The rate of reaction is Km. Then the enzyme substrate complex formation. Then inhibitor binds to binding site. Finally, we can get the enzyme substrate inhibitor complex. There no reactions will be takes place. Fourth one, reversible mixed inhibition. Mixed inhibition is the combination of two different types of reversible enzyme inhibition, either competitive inhibition and also uncompetitive inhibition. The inhibitor can bind to either the free enzyme or the enzyme substrate complex. In mixed inhibition, the inhibitor binds to site different from the active site where the substrate binds. This hypercatalysis in the enzyme substrate complex, which decreases the Vmax value. The diagrammatic representation, the enzyme which has the both binding site, inhibitor as well as substrate binding site. The inhibitor and substrate bind independent of each other. Either or both may be bound at any time. Completion of this reaction, however, depends on enzyme undergo a conformational change that leads to blocking the enzymatic activity. Comparison of these three competitive reversible inhibitors. Competitive, second one is non-competitive, third one is uncompetitive. The diagrammatic representation, substrate and also inhibitor compete with each other. Any one will binds to binding site, active site. Non-competitive inhibition, substrate binds to enzyme, inhibitor also binds to inhibitor binding site. Third one is uncompetitive inhibition. These type of inhibitors only bind with enzyme substrate complex, not for free enzyme. First one, competitive inhibition. I binds to free enzyme only and competes with substrate, increasing the substrate concentration overcomes inhibition of inhibitor concentration. Second one is non-competitive inhibition. Inhibitor binds to free enzyme or enzyme substrate complex, increasing the substrate concentration, cannot overcome by inhibitor inhibition. Third one is uncompetitive inhibition. Inhibitor binds to enzyme substrate complex only, increasing substrate concentration, which favors to inhibition of inhibitor. Next, move on to irreversible inhibition. Irreversible inhibitors are those that combine with or destroy a functional group and enzyme that is essential for its enzymatic activity. This inhibitor dissociates very slowly from its target enzyme because it becomes very highly bound to the enzyme either covalently or non-covalently. So that called as irreversible inhibition. The equation enzyme reacts with substrate to form enzyme substrate complex. Normally we can get the enzyme plus product, or enzyme substrate complex reacts with inhibitor to form enzyme substrate complex, enzyme substrate inhibitor complex, or enzyme directly reacts with inhibitor, enzyme inhibitor complex. Both are dead end of complex. Irreversible inhibitors are bind via covalent linking to the enzyme, causing modification of the enzyme and inactivating the enzyme. Many enzymes containing SH or hydroxyl or carboxyl groups as part of the active sites. Any chemical which can react with them act as an irreversible inhibitor. Example, heavy metals Ag plus, Hg2 plus, Pb2 plus have strong affinity for SH groups. Next, move on to types of irreversible inhibition. Group specific covalent modification agents, affinity labels, transition state analogues, suicide inhibitors. Example, group specific covalent modification agent. This type of inhibitors reacts with specific type of enzyme functional group. Example, serine hydroxyl group, cysteine SH group, or histidine imidazole group on enzyme protein. Example, diisopropyl phosphofluoridate DIPF, potent nerve gas poison which reacts with specific reactive serine hydroxyl on many enzymes such as catalytic hydroxyl group of acetylcholine esterase at synaptic junction which modifies the inactive form. Enzyme will be inactivated. Acetylcholine esterase which has the hydroxyl group, DIPF reacts with acetylcholine esterase which releases the fluoride and also protons. Finally we can get the inactivated form of acetylcholine esterase enzyme. One more example, irreversible inhibition. Chymotrypsin which reacts with diisophosphoprolyl fluoridate which releases the fluoridate DIP chymotrypsin as a inactive form. Diisopropyl phosphofluoridate is an irreversible inhibitor of enzyme chymotrypsin. Thank you.